So let's talk about stem cells in medicine, right? Stem cells are really just cells that can differentiate to different cells and then become different cells, right? So here you see a stem cell which can differentiate to become a muscle cell, fat cell, bone cell, blood cell, nervous cell, epithelial cell, immune cell, sex cell. And if you have a stem cell which can differentiate to all 200 somatic cells in the body and other, non-somatic cells, well then that you call totipotent. They can differentiate to all sorts of cells. Now pluripotent, which is right here, can differentiate to almost all cells. Multipotent can only differentiate to, for example, blood cells or neutrophils or something like this. And then oligopotent less, and then unipotent, they can only differentiate to one cell. So about stem cells is, firstly, they divide by mitosis and they differentiate and become more specialized. These are the two main things you need to remember about stem cells. Now I want to say here that I think one very good example you guys should know is a multipotent hematopoietic stem cell, which can give rise to all sorts of blood cells. So remember this one, we have a multipotent hematopoietic stem cell, and this can give rise to all sorts of blood cells. And therefore we also utilize this stem cell to treat various blood disorders, right? This can also be used in bone marrow transplantation to treat various blood disorders, leukemia, types of anemia. And the transplanted stem cells can repopulate the recipient's bone marrow and restore the normal blood cell production. Also, this is used in gene therapy. This multipotent hematopoietic stem cell, for example, in thalassemia. Remember, thalassemia is abnormal production of hemoglobin. To treat that, we also utilize a multipotent hematopoietic stem cells to treat a severe combined immune deficiency, in short, SCID. So please remember that multipotent hematopoietic stem cell, which can give rise to all sorts of blood cells, we utilize this specifically to treat, for example, thalassemia, SCID, other various blood disorders, leukemia, types of anemia, so on and so forth. Because you need, I think it's good to remember, some examples here. Anyways, how did we discover stem cells? Well, first of all, as we've just pinpointed, stem cells is in fact just cells that can differentiate and become more specialized. And I want you to understand that inside our body, whenever our tissues are damaged, well, these tissues get repaired and replaced by other cells. And so we actually do have a lot of stem cells in our body, right? So stem cells are normally found in the body, very important to remember. So in fact, Robert Hooke in 1965, in theory, he discovered this, right? When he discovered the cell. However, it was only much later until they understood, stem cells in depth. And firstly, hematopoietic or multipotent hematopoietic stem cells were discovered in 1961 by James Till and Ernest McCulloch in bone marrow, which give rise to types of blood cells. And then later on, 20 years later, Martin Evans and Matthew Kaufman successfully isolated embryonic stem cells from mice, and they saw that these could differentiate into all types of cells in the body, making them pluripotent. Remember, totipotent and pluripotent, these are in one category known as embryonic stem cells, found in the early embryo or zygote, and can differentiate to all sorts of specialized cells. And then adult stem cells is those found in the bone marrow of all ages of people, which can differentiate into various different types of blood cells, and this is very hard to isolate. This is one example I give you, multipotent and hematopoietic stem cells. Anyways, so how did they do this? Well, the discovery stem cells were actually done when they research how long cells live, right? The half-life of cells, where the half-life of cells in different organs got measured, and they saw like, okay, well, if there is a half-life of different organs, well, that means that those cells need to get replaced. So there must be some cells which replace these cells. And then we came to know stem cells, right? And they did this by doing a pulse chase assay, where they labeled the cell for a short period of time, and then they saw, that they got replaced. So yeah. Now let's talk a little bit more about gene therapy. So before that, just remember, we extract stem cells, for example, the embryo, and we do ex vivo stimulation of these so that they get become specialized cells, and then we give them back to these patients to treat them for various things. So this is a very short understanding of how we use stem cells to treat patients for different things. Now, about gene therapy, we must first understand that we can talk about in vivo and ex vivo gene therapy. Right, so in vivo meaning in the body, and ex vivo meaning out of the body. So somatic cell therapy is alteration of genes in human somatic cells to treat a specific disorder. Essentially, if we're talking about ex vivo gene transduction, what we're doing is we're taking the patient's cells and are extracting them and manipulating them outside the body. So this is ex vivo gene therapy. Now, rarely in some cases we use in vivo therapy, where cells get treated while in the body. I do want to say here that good candidates for gene therapy should be easily accessible and also have a long lifespan in the body. And that is why we typically utilize cells in the bone marrow, okay, because they fill in these criteria. However, it is quite hard to isolate these. Anyways, so when we're talking about loss of function mutations that result in a missing gene product, we want to fill that gene product out. And so we can replace a missing gene product by inserting a normal gene into that somatic cell. And to introduce genes to cells, this is done via vectors. And the best vectors, remember, it's very hard for things, just to come inside the body, and then the specific cell, the plasma membrane, and then the nucleus. So the best thing we can utilize here is viruses, right? Of course, we make it so that the viruses are a little bit different. For example, they don't replicate and spread to other organs, and also that they don't, contain the viral gene, right? So the first thing I want to talk about is retroviral viruses, which is a form of RNA virus which can insert copies of its genome in the nuclei of host cells after reverse transcribing their viral RNA into double-stranded DNA. So this is basically it, but this is a very, very condensed, version of what we're doing here. But essentially, the retrovirus is coming inside the host cell. Now it becomes to single-stranded DNA, and now you reverse transcriptase to making it double-stranded, and then it comes into the nucleus via an enzyme, and then that gets inserted into the gene, right, replacing the missing gene, and now you should be good to go, right? Now, I do want to say the disadvantage of retroviruses is that if they integrate near promoter regions, they can get inserted close to a proto-oncogene and activating it so that it becomes an oncogene and initiating cancer. And this is very problematic. And I'll make a part 2 series about this, so just Keep on watching.